Hi, good morning, everyone. Thank you so much for being here. It is my pleasure to introduce Dr. James Morton. For those that have not had the chance to look up his impressive Google Scholar, he's at the Flatiron Institute, which is a private foundation that funds research on the mathematical and theoretical sciences, computer science. And before that, he was at UC Davis, where he did his PhD with Dr. Rob Knight. He's been actively involved on the development of the CHIME software for analysis of microbiome data. And he's really in tune with the best practices going forward in terms of the best ways of normalizing our data. So, Jamie, thank you for being here.
Well, thank you for having me here. It's been really exciting seeing the kind of work that's been coming out of Penn State. So in my talk, I'm going to be focusing on how can we make sense of these microbiome data, and specifically coming from a more statistical angle. My focus is developing statistical methodologies and trying to figure out what sort of questions we can and cannot answer. Before I begin, I do want to acknowledge a couple of key people that have been fundamental and enabling this sort of work. So first, my PhD advisor at UC San Diego, Rob Knight. And I also want to acknowledge Peter Dornstein and Carson Zangler, who enabled the work I'm presenting today with the multiomics and the differential abundance work I'm going to be presenting. I'm now working at the Flatiron under Rich Bonneau, and as Erica mentioned, it's a private foundation funding mathematical research in multiple disciplines from physics, astrophysics, as well as biology. And in his group, I'm continuing to look into modeling microbial communities, their dynamics, but also looking at the functional abilities of microbes and from their genomics as well as their proteins. I won't discuss that for this talk. I'll be talking about the work I've been doing at UCSD. Okay, so let me first give a little bit of context why we're trying to understand these microbiomes in the first place. You see, there are more microbes on Earth than there are stars in the observable universe. And of these microbes, we can culture less than 1% of these microbes. But over the last 10,000 years, we've used them to culture our cheeses, to make our beers, to produce our drugs. There's a very long laundry list of things we've been able to accomplish. And one thing I like to think about is, well, what if we could harness, say, 10% of the microbial biomass on Earth instead of just the 1% that we're accustomed to? And with high throughput sequencing technologies, we might be able to edge a little bit at this question a bit more. So with DNA sequencing, we can take samples, and we don't need to culture those microbes anymore to observe them. Now we can directly observe their sequences and observe the distribution of microbes across different environments, whether it be poop, soil, marine, or so forth. And with the sequencing data, this enables a whole new breadth of possible questions that we can ask and answer. The typical types of questions that you might see are, well, one, can we identify if there is a difference between the microbiome? If I just look at these two mice, it doesn't require any fancy machine learning to say that this one's a skinny mouse and this one's a fat mouse. But if you can ask if there's a difference in their microbiomes, well, that could be interesting. And that can start to yield some mechanistic insights behind what the obesity phenotype. So one, can we identify if there's a global difference between their microbiomes, the microbial communities in their gut? If we can establish that there is a difference, then we might want to ask, can we identify the specific driving microbes, the specific microbes that are responsible for the shift between the obesity and the non-obese phenotypes? And this is the problem that I'm going to be focusing on the first half of my talk. But before I dive into the specifics, I do want to acknowledge that in light of the amazing discoveries that have been made in this field, there have also been a bit of pushback and skepticism about what we can learn from microbiome. And Nature went ahead and actually collected a survey across the biological sciences, not just microbiome, because reproducibility is an outstanding problem across the biological sciences. And to show how hard this problem is, even this survey was not reproducible. So they actually had to issue a correction to this study. So reproducibility is hard. So here's a basic outline of tasks that I'm going to talk about. The first half of the talk will be about differential abundance and the fundamental problems behind it, what we're interested in and what we can try to answer. And then the second half will be talking about applying some of those techniques into analyzing multi-omics and merging multiple data sets together. Okay, so, we all are collecting building experimental designs and collecting cohorts in some shape or form. We, for instance, if we want to say, okay, is there a difference between these two phenotypes? I would take multiple samples from multiple people. And if I want to say if there's a difference between sick and healthy, I have two cohorts. If I can establish a difference between these two cohorts, then I would hope to be able to identify microbes associated with the healthy cohort, microbes associated with the disease cohort. And maybe then I can follow with experimental validation and gain further mechanistic insights. So this problem of identifying the specific microbes that are key to this system is actually a very difficult problem. So there have been multiple papers coming out over the last decade showing how high the false positive rate is, where tools will identify a specific microbe to have changed across condition and enriched and specific phenotype. But in fact, most of those are actually wrong, that they're false positives. And one paper here showed that it's not possible to control the false positive rate. And we showed in one of our papers that that's correct, that the false positive rate can actually go upwards to 100% in certain scenarios. So to really understand why this is such a problem, let's try to take a very simple example. And just imagine for a moment that we are ocean microbiologists, and we're interested in counting microbes in the ocean. Now, I cannot sequence all the organisms in the ocean. We just do not have the infrastructure for that. But I can take a scoop of seawater and I can try to count the microbes in that scoop of seawater. So in this particular example, the entire size of the ocean is hidden. I don't know the total biomass, but I do know that there are 6 orange microbes, 6 blue microbes in this scoop of seawater that I collected. And let's say that there's been a disaster and BP strikes again. And now we take, and the beach is destroyed, and now we're like, okay, well, what happened to the microbial ecosystem after BP came into the picture? So let's say that, okay, I take another scoop of seawater. Now there are 8 orange microbes and there are 4 blue microbes. What happened between these two scenarios? Did the blue microbe decrease? Did the orange microbe increase? Or was it a mixture of both? Okay, so let's take another look at this picture. Let's say that the entire ocean only has 24 microbes in it. And one possibility is that only the blue microbes decreased. So I've dropped from 12 microbes down to 6 microbes. The orange microbes stayed the same. But you can see from my scoop of seawater, I have 6:6 ratio here, 8:4 ratio here. And that the total microbial load, the total population of the ocean has strictly decreased. But there's actually another possibility, namely that the total microbial load actually increased, that the ocean bloomed and loved this oil. And now we still count 8 orange microbes, 4 blue microbes in this bucket of seawater, but everything grew. And so then the question is, well, what can we ask here? if you take a look at these two buckets of seawater, they're identical, but these are two very different biological scenarios. Then the question is, well, what can we infer based off the scoop of seawater if we can't differentiate these two scenarios? Okay, so that's the problem at hand here. So in order to get a good grasp of this problem, we need to go down to the fundamental math behind how to make sense of proportions. So the real problem is that in order to determine if a species has changed abundance between different experimental conditions, we need to know the total size of the community. Oftentimes it's very hard to determine. In some cases, we're lucky. If you have a petri dish and you have flow cytometry, maybe you can count the microbes in that petri dish. But if we're dealing with gut, marine, soils, this could be a very intractable problem depending on your system. So try to understand the fundamental mathematical problem here. Let's try to imagine we have two samples. We have here the absolute abundances after the event. Here are the total number of microbes in that environment, or I have D species, okay? And these are counts after the event happened. And then B denotes before the event happened. So these are the abundances of those microbes before that event. If we want to determine which species has changed, all we need to do is just take that ratio if we have the absolute abundances. So let's focus on species 1, for instance. If I want to say, okay, did species 1 increase in abundance or not? All I need to do is just take that ratio of A1 to B1. If it's equal to 1, there's no change. If this is greater than 1, then the abundance after the event is greater than the abundance before the event, is increased. And vice versa. If it's less than 1, it decreased in abundance. Okay, so now here's the real crux of the argument, is that we cannot observe A and B directly. We don't have access to the total of the absolute abundances because we don't know the total biomass of the system. So we can only estimate the proportions based off of the sample that we collected, and that the total biomass is hidden from us. So if we actually try to compute the changes that I just showed you, this is confounded by the total biomass here. That we have the total biomass shift before and after the scenario, and that this will confound our ability to infer if there is a change in the absolute abundances of these microbes. Okay, let me know if this makes sense. This is the punchline. Okay. All right, so that's the problem. You need the total abundances. So what can we do with our sequencing data? So there's two approaches that we propose. One is to use the ratios, which we also call balances, borrowed from the compositional data analysis literature. Here's our paper that discusses both of these techniques in detail. It just got published fairly recently. All right. So it's clear that we need a way to negate the total. Yes?
When you say the abundance, is it just a count or is it just on the biomass?
Good question. So these are the individual, so right here, when I mean the abundance, I mean the individual count of the microbes, but we're talking about in the entire ecosystem of interest. And what I mean by the total biomass, that means the aggregate, the total count of all the microbes together. So oftentimes we can't measure the actual absolute abundances of the microbes in the entire ecosystem, but we can try to estimate their proportions because we can count those microbes with sequencing. But the total biomass is much harder to estimate in many scenarios. That's a good question. Okay, so So okay, we need a way to kill this total biomass if we can't measure it. One possibility is to look at just ratios or concentrations. This is what chemists have been doing for the last 200 years. So I need to take, instead of just one chemical, I need two. Like if I want to look at salt, for instance, I'll take the ratio of salt over water, and here water is my reference. So hence the title of this talk, the reference frames. And so in this, and along the same analogy, we need to choose a microbe as a reference. So here I specified the microbe D to be that reference microbe, but every analysis will be different. This depends on what you choose. So if you can pick a reference, doesn't matter which microbe you choose, you'll notice that the total biomass will cancel out from the numerator, the denominator. And now from this, you can infer that the ratio of the proportions is equal to the ratio of the absolute abundances, because now we've canceled out the need to compute this total biomass. So if I'm going to that scenario I showed before, if I look at the ratio of orange to blue, and you can see that that all the scenarios I showed before will have the exact same ratio. So I can't tell the difference between those possible scenarios, but maybe that's just a limitation of my data. But I can tell you for certain that the ratio has shifted. And this is something that I can't answer with the data at hand. Okay, show that this actually works. We took a look at atopic dermatitis. In this particular data set, they took metagenomics sequencing, and they tried to count microbial taxa across different lesion sites. Furthermore, they looked at the multiple time points before this infection, this flare, and then after the flare. And they tried to see how much does this change? How much do the microbial communities change? Here, this axis denotes the SCORAD, which is a measure of the severity of the infection. In this study, they noted that Staph aureus was significantly changing across conditions. But then they noted in previous literature that Staph epidermis and M. globosa were noted in previous literature, but were not detected in this study. M. globosa is particularly interesting because it's piqued the interest of multiple researchers, but there's not a consensus of what's actually doing. Some researchers have concluded that it's increased between lesion and non-lesion sites. Others have concluded that's increased in lesion sites. Some concluded there's no difference. And in summary, there's little doubt that M. globosa plays a role. It's just not clear what it is. So we decided to take another look at M. globosa in this particular study. So if you look at the differences between the lesion and non-lesion sites, sure enough, there's no difference in the proportions. And if you look at these proportions due to the with regard to the SCORAD, the severity score, it's flatline. There's nothing significant there. Then we took a look at P. acnes. P. acnes is, in this system, the most abundant microbe. Our rationale is, well, because it's the most abundant microbe, maybe we can use this as a reasonable reference because it's everywhere. So that's what we did. We took the log ratio of M. globosa abundance over P. acnes, and sure enough, there's a significant difference between the lesion and non-lesion sites. And when we look at the SCORAD, there's a very strong correlation between these two microbes and the severity. And you might think, well, Jamie, this is statistics. This is maybe a one-off shot. Like, you just got lucky. Well, we look at another data set that just got published and looking at lesion and non-lesion body sites. And sure enough, we found the exact same pattern pop up. So we were actually able to reproduce this pattern across two completely different data sets. And we knew this pattern before we even looked at before this data set even got published. So that's one demonstration showing that looking at these ratios could be useful. But then the question might pop up, well, how do you choose a good reference? Especially with ecosystems I don't completely understand. So there's another technique that another perspective we developed looking at ranks instead. So here's a way to think about this. I don't know the total biomass shift, but I do know that that total biomass shift applies equally to every microbe. So rather than trying to focus on if the microbe has changed or not, maybe I can quantify some sort of relative shift. And what I mean by that is that if I look at the ordering of the fold change I estimated from the proportion, and I try to sort that list, that list of microbes will be in the same ordering as the absolute fold change, meaning that I can just sort the microbes according to how much they change, and that ordering should be consistent. Okay, so here's another way to think about it. Say that I have this is a distribution of microbes before the treatment. This is distribution of microbes after the treatment. I can divide each element-wise each microbe abundance. If I take the log of that, it'll look something like this. So the takeaway here is that I can infer which microbe has increased the most, which microbe has decreased the most, but I can't actually tell you if that microbe has changed or not. And here's another diagram what this will look like. So right here we have the log fold change of the absolute abundances, and then here the log fold change of the inferred proportions. Every dot here represents a different microbe. Every color here represents a different biomass shift. So here is a scenario where the change of the total biomass made it such that the inferred proportions will give you a lot of false positives if you're trying to infer which microbe has changed. But you can also get the vice versa. You can also get a lot of false negatives, depending on how much your total community has changed. But if you notice from here, everything is in the same order, meaning that if I just forget the intercept for a moment and just focus on how to sort these microbes according to how much they've changed, I can do that. And that their ordering is identical across all those possible scenarios. So, all right, how useful is this? Well, to showcase this, we looked at the oral saliva time series. This data set is particularly nice because we could actually quantify the absolute abundances here. The saliva has you can quantify the flow rate of the saliva out of the mouth. So once you do that, you can couple this with flow cytometry and then count the number of microbes that have been collected within a unit of time. So we can get some proxy of absolute abundance that way with flow cytometry. And here's the flow cytometry data looking at people before and after they brush their teeth. And you can see here that there's a dip in the total number of cells after you brush your teeth. Well, it's reassuring. Your teeth are not getting any more dirty. And the oral biofilm is particularly interesting. Right here, I'm going to highlight two microbes. Here are actinomyces. These microbes form at the root of the tooth. They serve as an anchor to anchor down the biofilm to the teeth. And then here we have Haemophilus. These are secondary metabolizers. They live on the periphery of the oral biofilm and basically on the surface. And one thing we can do is we can compute these ranks essentially by computing the log ratio of the average abundance of a microbe before brushing teeth and the average abundance after brushing teeth. So, and once we have these log ratios per microbe, we can sort them and try to see what pops up. Here on this end we see actinomyces that are heavily dominated on this side of the spectrum. which this makes sense because after you brush your teeth, the main players that are left are actinomyces, because those are the ones that are hardest to brush off. They're the ones serving as the anchor of the biofilm. On the other end, you have Haemophilus. These guys are going to be more prevalent before you brush your teeth because these are the first ones to go because they're on the top of the biofilm. So this matches the biology that we expect. And to show how this sort of methodology is compared to other tools, we did a quick benchmark. So the technique I just showed you to compute the ranks, in order to compute those averages, we use something that looks like a multinomial regression. And we compared this ordering to ALDEX2 and ANCOM, two prevalent differential abundance tools. If you squint really closely, you'll see that the outline of our multinomial regression we developed is similar to ALDEX2. Well, it turns out ALDEX2 underneath the hood also uses a multinomial regression. So this is a nice sanity check for ALDEX2 that the ordering somewhat matches what we'd expect. But here, if you take a look at ANCOM, note that everything highlighted in red is deemed to be significant. Note this is kind of weird because in this sort of environment, you expect everything to change, right? Like you don't expect anything to increase or decrease. Sorry, you don't expect anything to increase in abundance, but that's what ANCOM detects. So especially these guys are very weird. They should not be detected to be increasing at all. And furthermore, ALDEX2 didn't detect any significant patterns because what that is doing underneath the hood is it is comparing microbes to the reference, the average. So if your average isn't changing, ALDEX2 is fine. But if your average is changing, like in this scenario, then that's when things can go wrong. So it didn't take anything to change. So the ordering could be useful, but the statistical tests are not very trustworthy. So the conclusion with differential abundance is that differential abundance on the absolute scale is not possible because our data is relative. We cannot infer absolute changes from relative data. And as a result of this, over 100,000 papers that have been published in our area needs to be rechecked because they have possibly flawed interpretation regarding to this differential abundance problem. Now, this might be a curse, but it could also be a blessing. We could be, this could be an opportunity to learn some novel biology from the existing published literature. All right, I've got 20 minutes left, so that should be just enough time to cover the last part of the talk about multiomics. So, okay, so basically give it a quick refresher. You see, you can't forget that these microbes are essentially like chemical factories. They mediate chemical reactions and giving us the cheeses that we like, giving us the climate that we live under, and are responsible for many of these global processes. And with mass spec, we might be able to get at trying to estimate at the molecular abundances, including the molecules that we aren't very familiar with. So another question is, can we take these molecular abundances, can we take these sequencing abundances? What can we learn about the metabolism of these microbes? So here's a couple of challenges. I'm going to highlight the annotation rates are quite bad. So less than 2% of the metabolites are annotated, and even fewer of the micrometabolite products are documented. It takes about 1 PhD to annotate one of these interactions. So if we're going to make a prediction, we better be fairly accurate about this. Otherwise, that's a wasted PhD. Okay, so and really the best techniques out there are looking at correlation. If I look at the abundances of my microbes, do they correlate with the abundances of a metabolite? And I'm going to show, for a similar reasoning, why this can be problematic. So I'm going to focus your attention on just the dark colors here. So dark green metabolite here, the dark blue metabolite microbe here. In this particular case, there's a very strong correlation between the dark green, dark blue abundances. But if we look at the proportions, like you can see that that pattern is no longer apparent. This is an example of a false negative. False positives can also happen. If I look at the same dark blue microbe, dark green metabolite, there's no relationship in their abundances in this particular system. But if I look at the proportions, you can see that now there's a strong pattern. This is a false positive, and where this is a spurious hit. And the real problem is that correlation is not well defined for proportions. And there have been multiple papers that come out over the last decade showing why this is the case. And Pearson himself, 100 years ago, said don't use Pearson correlation on proportions. And you know, there's still quite a bit of confusion surrounding this issue. So the approach that we're trying to take is, well, maybe we don't want to look at the correlation. Maybe we can take a look at another metric. For instance, co-occurrence. So why co-occurrence probability? Well, one, it has a natural probabilistic interpretation. For a given microbe and metabolite, the co-occurrence probability is given by here. The probability of observing a metabolite, given you've already seen the microbe. If you can identify this, and maybe that's good enough to get a good foothold about which metabolites could be associated with a given microbe. And the best part about this is actually tractable. We can actually learn this from these types of data sets. To try to wrap your head about how these co-occurrences can be calculated, we took a look at word2vec. So this is a machine learning algorithm commonly used in natural language processing. So the way that this algorithm works is it takes a look at many sentences, and then given a single word, it tries to predict the presence of other words in that sentence. So if I take this joke right here, and I basically pick out one word here, then the algorithm will try to take this word as input and then try to predict other words by context. If I train enough of these dumb jokes, then I can be able to learn some sort of grammatical structure. For instance, maybe the word people commonly co-occurs with other words like who. People who like to, you name it. So once you build this algorithm and it can try to predict these co-occurrences, now you can start asking some pretty interesting questions. Well, for one, you can start to map each of these words to a coordinate system. And you can ask, for instance, what is the distance between king and queen? And is that distance equal to the distance between man and woman? Now I can do something that looks like analogy reasoning. If I know the coordinate for king, and I know the coordinates between man and woman, then I can from that try to compute the coordinates of queen. So then I can infer that king to queen has a similar relationship to man to woman. And the NLP people have been doing this for a while, and a lot of interesting analogy reasoning tasks have been solved using this sort of approach. So what we've done here is we basically took this idea and then we basically swapped out the data types. So rather than trying to predict, input a word, predict a word, here we're trying to input a single microbe sequence. From that microbe sequence, trying to predict the metabolite abundances for all of the metabolites in that sample. And once we can iterate this model across all of our samples, then we can do two things. We can learn coordinates for our microbes and for our metabolites, and we can learn these co-occurrence probabilities. Given the presence of a microbe, can we predict what metabolites would be found in the presence of that microbe. Okay, to show this actually works, we took some simulation benchmarks using differential equation model of a cystic fibrosis biofilm. So I'm going to walk you through this. Over here on the x-axis, we've got top K hits. These are the top K most associated metabolites for a given microbe. So basically, we're trying to look at how good are the most predictive metabolites. And here we're looking at precision and recall. You can think about this as the false positive rate, the false negative rate. How many metabolites can I correctly identify associated with a microbe? How many metabolites will I correctly miss? Okay, so you can see right here that our tool MMVec is pretty stable. Here's random. And notice that almost all the tools are underperforming compared to random, meaning that the most commonly used tools, Pearson, Spearman, you name it, is worse than randomly flipping coins. This is not great considering how expensive our data sets are and how much love and care was given to create these data sets. We need to outperform random. Over here, but you can see this scale is actually between 0 and 1. This is a hard problem. We're operating at 1/4 of our potential. Now there's still much more room for improvement, but we're beating random. So we're starting somewhere. So okay, I know there are a few soil scientists in this room, so maybe you guys can appreciate this. So here we looked at desert biocrust soil. In this particular experiment, they're interested in looking at how the soils respond after a wetting event. So they took multiple time points, and in their paper they concluded that microcoleus is one of those colonizers, and that creates the byproducts required for other microbes to colonize later on. So the really nice thing about this particular data set is they were actually able to get validation data. So they collected the samples from the deserts, but they've also cultured these microbes and then ran mass spec on the individual culture, so they can get much more certain about what byproducts were actually produced by a given microbe. So over here, all the solid arrows are the micrometabolite interactions that were consistent between the experiments in the lab and the ones that were collected from the field, the sequencing mass spec data from the field. Notice that there's a lot of dotted arrows. These denote interactions that were contradictory, but that were detected in the lab. And if we look at this particular microcoleus, half of the interactions were contradictory between the experiment and the survey, the samples that collected from the field. So we did another benchmark, taking a look at MMVec, our tool, compared to the other tools commonly used. And sure enough, the true positive rate of MMVec is much higher than the existing techniques. And you can see Pearson and Spearman barely detect any of those interactions. Let's see. Okay, I have some time left. Okay, okay, now I know many of you use machine learning techniques, and you're probably familiar with these techniques being black box solutions. So one thing we wanted to do is to try to open up the box a little bit. How can we actually interpret what the model is actually learning? What does it actually mean? So one thing that we realized was that the algorithm word2vec is essentially like PCA that we commonly like to use. And because it is basically PCA, now we can throw all the ordination techniques that we love to use and use those techniques to make sense of these models. So here we have what's called a compositional biplot. I'm directly trying to infer the co-occurrence patterns of the microbes and the metabolites. So let's step through this carefully. Every dot here represents a metabolite. If two dots are very close to each other, that means they're tightly co-occurring with each other. Every arrow as a microbe, same sort of rationale applies. If two arrows are close together, that also means those two microbes are tightly co-occurring with each other. From a high-level overview, you can see that there's two types of microbial communities here. There are pathogenic microbes on this side highlighted in green. There are anaerobes here highlighted in orange. There's a lot of names here, so I'm going to focus your attention on just one of them for now, namely the Pseudomonas aeruginosa. So this guy's a known virulent factor. It's a virulent microbe in cystic fibrosis, and it's been very well studied due to its potent capabilities. So that's the red arrow here. And there are all these molecules I highlighted in purple. These are rhamnolipids. These are molecules that are known virulent factors that are known to be produced by Pseudomonas. And you can see that arrow, the Pseudomonas arrow, going right through that cloud of rhamnolipids, which we know is being produced by that specific microbe. We can dissect these sorts of models even further. I can take the first principal axis for the microbes, and then I can try to see how those microbes divide up with regard to the variation of, okay? So here I've focused on the Pseudomonas aeruginosa. And Pseudomonas aeruginosa is the most abundant microbe in 40 samples. And Streptococcus is the most abundant microbe in 8 samples. The rationale is if they're the most abundant microbes, maybe the metabolite profiles in those specific samples will reflect what the abundances of those microbes look like. And sure enough, they do. So while we can go back to the raw data, given our model, we can pull out the Pseudomonas abundances and the Streptococcus abundances. Sure enough, they perfectly co-exclude each other. They are never seen in the same samples together. And then we can then take the exact same samples, take a look at the metabolites, and sure enough, the metabolites almost perfectly co-exclude each other as well, as predicted by the model. And when we're looking at the top 100 metabolites associated with Pseudomonas, and the top 100 metabolites produced by Streptococcus. So this paper is almost out. It'll be out in a couple weeks, I hope. But feel free to follow up. Everything is available. It's on GitHub. And of course, you can email me with questions later. And I do want to acknowledge that this was not a lone rogue project. This was a very large group effort from very talented biologists and as well as computer scientists and statisticians. And I want to especially highlight Lisa Marotz, whose in-depth biological insights really enabled the first paper I presented looking at atopic dermatitis and the oral time series. And I want to thank Alex and Justin. Justin, who's now going to be joining Penn State. Their statistical insights were really key for making this story really pieced together. And finally, I'd like to thank Rob Knight, my PhD advisor, and for really enabling this sort of work. And thank you guys for listening. Yes.
So you had the plot where you showed the Pearson and experiment correlations being worse than random. So that says that there's a bias towards being wrong in some way. So can you explain sort of like what that is or how that's happening?
Yes. So the reason. So why do we think this is the case? So basically, this all boils down to the total biomass. We can't measure the total biomass, and because of that, we can't quantify what's actually changing. Pearson experiments don't account for this, and because of that, they don't work very as predicted. So the only scenarios that Pearson experiment will work is if the total biomass is constant. If you have on a closed container and total number of microbes doesn't change, those tools will perform as expected. But if the biomass changes, those tools will not perform very well.
So
Yes.
in that case, you can't have any bacterial growth, right? Let's say you have 2 jars of fermentation, and if the fermenters grow there, then Pearson correlation is not going to work.
You can't have.
It's a closed system.
Exactly. You can't have total microbial growth. So if the total number of microbes in that system changes, that violates the assumption of the Pearson experiment when applied to proportions. Those tools are not designed to deal with proportions. Yes, and then you.
The co-occurrence, are you defining that based on concentration or presence absence?
Good question. So both actually. The way that we build this algorithm is that we base it off the presence of a microbe. So, but the way I define presence is actually a little different. I'm doing a sort of bootstrapping technique, where I have my entire microbial population. I randomly pick one microbe, one sequence, and I look at that particular microbe. I treat that as present. But then I look at the relative concentrations of the metabolites. I do want to note that the metabolites also have this problem too. We don't know the total mass of the metabolites as well. So we look at those metabolite proportions. And that's how I can try to etch at this sort of relationship.
Thank you.
Very impressive job, James. That first question regarding your first part. So how do you deal with the inflated zeros?
Yes.
Right?
Okay, zeros are a problem, and I totally breezed through that. The way we did it is using the multinomial. Yeah, that's a good question. Oops. Yeah, I kind of hid the math in this presentation. It's all hidden here. The multinomial model has zeros built into it, so you can deal with that through that way. It's the simplest counting model that you can use in order to model how to count microbes in a bucket.
So my second question, if you conduct your differential abundance in the microbial wide scale, so in the broken promise paper,
Yes.
The classic Benjamin and Hunsberg FDR control procedure fails partially because of the negative correlation imposed by the compositional nature.
Exactly.
The procedure, if you divide everything by a common reference level, that will introduce a correlation among those individual differential abundance tests. Okay,
good. Okay, these are all good questions. I'm going to be honest. I don't have a great handle on FDR correction at the moment, and I think this is an open-ended question right now. Yes. So I think if you're doing the post hoc tests with the reference, you need to be really careful about what that correction scheme will look like, because now you're looking at possible references as well. So I'd love to talk in more detail about that. The answer is I don't know, but this is something that I think needs to be addressed.
Job security.
Yes.
This new method that's about to come out was super awesome. I'm curious about the limitations of it. So both, I'm sure like on the rare side, if you have a lot of zeros, it's just difficult to look at co-occurrence between microbes and metabolites. But also on the opposite end, if you have a metabolite that's very widely made by a lot of microbes, do you have the power in your method to detect that? Or are you really finding like micro-metabolite connections that are very unique to a certain clade of microbes?
So, okay, let me piece through this. We don't know all the limitations now. We know a number of them. Now, if we have if one metabolite is being produced by all the microbes, that will probably have a high co-occurrence probability with everyone. Now, note that when we're doing this analysis, we're not looking to see if the metabolite is being produced by a microbe. We're just seeing if they're commonly co-occurring together. I'll give you an example. In our paper, we found bile acids co-occur with Klebsiella. And if you look at this at first glance, you might think, okay, Klebsiella produces bile acids. Turns out Klebsiella is actually resistant to bile acids. And because everything else in that community just died once the Klebsiella invaded, only Klebsiella could co-occur with those bile acids. So those confounders will definitely exist, and I think this is where biological insights is really necessary. The only thing that this tool does is try to help you narrow down the hypothesis space. Yes.
So for your two kind of methods, the reference frames or the ranking differentials, is there situations where one would be preferred, one method would be preferred over the other?
That's going back to the statistical question. So with the ranks, I don't know of a way to do statistical tests with that. I don't know how to test if the ordering is good or not. With the reference frame, now you once you got a reference, now you can actually begin to do statistical tests. So you can apply the standard t-test, the standard correlation on that ratio, similar to the way the chemists operate with the concentrations.
So what is the scenario where you would use the ranks then? Just tell if the two populations are the same or different overall?
Well, the scenario would be when do you know what a good reference is? Like with chemistry, everyone uses water, but in our case, is that a reasonable reference? And like, what microbe would we want to use? And that is a question that's outgoing. How to choose good references to enable statistical testing.
Have you played at all with like having spike-ins as your reference? Like, is that a more reliable?
That's a great question, and I think this will require a very careful conversation. My takeaway is that if you're looking at if you only care about your samples, that can help make the abundances comparable. There's technical challenges, but let's say you solve them, that could help. But when you want to start to make predictions across beyond those samples, I don't know of a good way to handle that at the moment. But I think spike-ins can help in some scenarios.
Let's thank James for coming. And we'll talk right up